So for our last biomolecule, I want to talk about nucleic acids. And so we're going to just jump right into, what is it, Handout 2J. Handout 2J is going to give us some basic information about nucleic acids, otherwise known as, DNA, RNA. Now, I don't want to get too detailed on this because, there's an entire genetics course and we just don't have the time to delve into this. But if you're really interested in DNA, you should look into the genetics course for sure. But just to give you an overview, up here on the top it says DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. The difference between the two is the sugar. If you notice, there is a sugar in the name. Ribose is a sugar. And in RNA, the ribose has all of its atoms. But in DNA, it's missing, this ribose sugar is missing an oxygen. So that D part of the name, deoxyribose, means that we're missing an oxygen off of that ribo sugar. And this makes all the difference in the world because DNA ends up forming this double helix and it holds our genetic material. We'll talk a little bit about RNA after I explain DNA, but RNA is a different molecule. It's only a single strand, and it doesn't carry our genetic information. It carries, well, essentially the code of our genetic information. And so let's look at DNA. Just ignore RNA here for a minute, and let's look at DNA. So DNA is made up of our famous double helix ladder. And it actually has, here at the bottom, I know it's small, so I'm zooming into it, but it has three components. Let me get rid of my little thing here. Okay, there we go. Now we can see better. I think it was on for the entire other video. Anyway, our DNA is made up of these structures called nucleotides. And so each nucleotide has three components to it. It has a phosphate group and that 5-carbon deoxyribose sugar. That makes up the sides of the ladder. And then it has this nitrogen-containing base. And this nitrogen-containing base makes up the rungs or the steps of the ladder. Now, this nitrogen base is what holds our DNA, and there are 4 of them. And so I have them listed right here. There are 4 bases: adenine, thymine, cytosine, and guanine. It turns out that adenine can only bond to thymine and cytosine can only bond to guanine. So that's what I have right here. So A's and T's and C's bond, and G's and C's and C's and G's bond. Well, they bond right here. So if we have cytosine, then it bonds to guanine. And it makes up the steps of the ladder. Now, these bonds are held, or actually I should say these bases are held by bonds called hydrogen bonds. And these hydrogen bonds are really weak. So the DNA can be unzipped fairly easily because these hydrogen bonds are what hold the bases. So what we can do is we can unzip them and split them apart so that we can read the DNA. And so DNA is easily copied, but it's also easily degraded. It's easily broken up. And that's why it's hard to get DNA samples off of a specimen that has been dead for a long time. There may be no usable DNA. And you may have heard that, in archaeological or paleontological, I can't say that word, explorations, as well as like criminal justice and stuff. RNA is a single strand, so it's just one single strand. And the big difference is that not only, it has this extra oxygen, but RNA doesn't have thymine. It has uracil. So uracil replaces thymine in RNA. And you can always tell the genetic code is RNA because it won't have T's in it, it'll have U's. And so this is just the basics of our nucleic acids. All right. So in the next following couple of videos, we're going to use nucleic acids, DNA and RNA, to talk about how our genetic code builds our body. In other words, all of these genes that we have are going to make proteins. And so we're going to cover that in the next few series of videos. Okay, that concludes this video. So look for the next video, which will be DNA replication. And then we'll talk about how we're going to make proteins through transcription and translation. All right, see you soon.